Okay, good morning. Okay, so today we're going to talk about DNA hybridization and some uses of it. Last time we talked about DNA melting, which is obviously important, but is not nearly as useful as the hybridization idea, as we'll see in our examples. So a reminder about last time, and the same conditions are pertinent to the hybridization reverse part of the melting reaction, okay, that conditions of the DNA solution will alter the melting temperature of the DNA. So high temperature, of course, favors strand separation, and there are several words for this. Dissociation, denaturation, melting are all the same. Now we know that that makes sense because non-covalent bonds are easily broken. Okay, so other conditions that affect the melting and hybridization are salt, right? High salt favors the strands staying together for the simple reason, well, not so simple, I guess, that it neutralizes the negative charges on the phosphates, thereby removing one of the things that drives the two strands apart. So high salt favors hybridization and favors the strands staying together, and so high salt will raise the melting temperature. Solvent effects also, some small molecules, contribute to the melting temperature and hybridization conditions in that they compete for hydrogen bonds. Okay, those include Formamide and urea, and these are commonly used in experimental situations. Okay, just a reminder from last time, if we look at the figure on the right, it says what we need to know, that the lower salt lowers the melting temperature, higher salt raises the melting temperature for the reasons that I just said. Now, GC content obviously also still comes into play, and it will come into play with hybridization as well. The GC base pair being stronger, the melting temperature will increase as the GC content goes up. Okay. So our formamide and urea, remember, these compete with the hydrogen bonds and therefore destabilize the double-stranded DNA structure, causing it to melt at a lower temperature. Here's a picture of our base pairs again. We've got formamide and urea, and they are able to form hydrogen bonds with the bases, and those compete with the real base pairs, thereby favoring denaturation and therefore lowering the melting temperature. Okay, so for today, we need to think about the reverse reaction, that of putting two DNA strands together. So our starting material will be DNA that's in its single-stranded form, that can now find each other in solution. They can float around and then hybridize if they find their complements in solution, hybridize to them. This is also called annealing. Okay, this is a very, very important process, as we'll see, for things including PCRs and sequencing, etc. Okay, so these are comparable considerations to the melting you want, except with some additional considerations, as we'll see. Okay, you want the correct perfectly matched molecules to hybridize, but you don't want the similar but non-perfect hybrids to form. And so you can imagine if you've got these DNA molecules floating around in solution, they might form some base pairs over short distances, but not be truly complementary to each other over their entire lengths. So you want to get things set up so that you get perfectly matched double-stranded hybrid molecules and not allow those other non-specific interactions to take over. Okay, so the key concept here is going to be stringency, or harshness, and that stringency will dictate whether the imperfect molecules form and also whether the perfect molecules can still form. Okay, stringency. Okay, for example, if we have conditions of high salt, this is going to be low stringency relatively because we can allow some imperfect matches to form. Okay, high salt, clearly low temperature will favor non-specific interactions and no formamide or urea. So these kinds of conditions allow imperfect hybrids to form, which is something people sometimes want, but that's not what you would want normally. Okay, so in terms of things that favor melting or destabilizing, those would be high stringency. So lower salt and high formamide would be a high stringency situation in which the hybrids have to be perfect to stay stuck. Let's reminder. For each of the following, will a change increase, decrease, or not change the melting temperature of a DNA molecule? Adding more sodium chloride to the solution might increase, decrease, or not change the melting temperature. Adding formamide can increase, decrease, or not change the melting temperature. Increasing the concentration of the DNA might increase, decrease, or not change the melting temperature. Okay, so now we know that adding more sodium chloride will increase the melting temperature because it favors the strands to stay together. Adding formamide decreases the melting temperature because it helps them fall apart, and the concentration of the DNA has no effect on the melting temperature. Which of the following represents the most stringent conditions for formation of DNA hybrids? Low salt, low formamide, low salt, high formamide, high salt, low formamide, high salt, high formamide. Okay, so the most stringent is the harshest possible conditions, and that would be the low salt, high formamide. So the many, many uses of DNA hybridization. Okay, there's a lot of more complicated things that are happening if you think about these two molecules floating around in solution trying to make base pairs with each other. So it's a more complicated situation mechanistically, really. So they have to find each other and then base pair correctly, and it's a fairly complicated and cooperative process. Okay, so the issues, if conditions are too mild, you could have incorrect base pairs forming. Conditions that are too stringent, the hybrids will melt away as soon as they form. So you want to have something which is perfect, if possible. Okay. So one thing that really is going to be important for this is that most of the time when we're trying to anneal two DNA molecules, we're not just taking genomic DNA from an organism and then asking it to find its own homologous complements. What we're doing instead is using small molecules, synthetic oligonucleotides, to try to find their complements in a complex DNA such as a genomic DNA. So the synthetic oligonucleotides are small and they're artificial. They're kind of fake in many ways because they are made by machine. They're made single-stranded, and they are relatively short compared to what you would see in a normal genomic DNA. 20 to 50 nucleotides is typical. So they can also be fake in the sense that they can consist of sequences that don't exist in nature. You can make any sequence that you want by machine, even though it's not necessarily present in the genome of the cell that you're studying. So examples of use are probes to detect complementary sequences, as in the example we'll see, but also used as primers as parts of reactions like PCR and DNA sequencing. Okay, so in contrast, genomic DNA from a person, if you think about it, that's a real DNA. It is fully double-stranded, and the molecules that are present even in a fairly chewed-up mixture are very, very long, several thousand or hundreds of thousands of base pairs long. So they're very complex sequences. By contrast, a rather extreme difference, our short synthetic oligonucleotides are single-stranded in the first place, and also typically in the range of 20, not base pairs, 20 nucleotides. They're not base pairs, they're single-stranded. They're also made by machine, not DNA polymerase, and they are not made using a template. It's completely done chemically. Okay, but they are single-stranded, not double-stranded, and they are totally artificial. Okay. The other issue that comes into play, and this can be important too, when you're taking a short oligonucleotide, 20 nucleotides in that range, to a genomic DNA, is the fact that they are short alters the way that they melt somewhat. Okay, the GC content is still important, but the length does come into play, and that's partly the result of a phenomena called DNA breathing, where the ends of the molecules will fray and sort of start opening up spontaneously, even under very mild conditions. And that's shown in this picture here, where we have the molecule on the left is base paired along most of its length, but at the ends have started to sort of come apart. Now that can continue to come apart, or it can zip itself back up again, as shown in the right part of the diagram. Okay, so breathing does occur spontaneously, internally, but also at the ends. So for example, in the picture on the left, we have typically a AT-rich region will locally sort of melt into what's called a bubble, where it's locally disordered, but can go back into its normal ordered condition. Okay, in the process of melting, you start off with these bubbles, which can then take over the molecule and cause strand separation. Okay, and the one on the right is close to what I showed you last time, where you have the end of the molecule kind of opens up and can zip up back together, but can also favor actually the whole molecule falling apart. And this comes into play mostly with short nucleotides, well, short double-stranded molecules, much more so than with long genome length ones. Another phenomena which is really important for small molecules that we're going to be using for our hybridization is that they are very sensitive to single mismatch along their lengths. Now, this is an experiment that was done to sort of look at what happens if you have mismatches. So what the people did, they designed a molecule that was 16 mer, which is to say it's 16 nucleotides long, and they made that molecule. And then they made the complement of that molecule, which was an exact match to the 16 mer. Then they took one of those molecules and systematically changed one nucleotide at a time. So they had a whole collection of 16 mers, which matched the first one at all positions except for one. So there would be one position of mismatch within this 16 mer. And the question being asked in this experiment was whether the placement of that mismatch has some effect on the ability to form stable hybrids with the complement, or almost complement. So that's what's shown in this diagram, this graph, that the mismatch base position is shown on the left. So nucleotides going from 1 to 16 is from left to right on the x-axis. And essentially, the fraction hybridized, as measured by an absorbance, is shown on the y. So a fully base paired or exact match oligo will, of course, base pair very nicely. And even molecules that have a single mismatch close to their ends don't do so badly. But what happens when you have your mismatch smack in the middle or close to the middle of the oligo, that drastically destabilizes the hybrid, causing them either not to form or to melt off very quickly. So even a single mismatch within a 16 mer can have a drastic effect on whether the molecule can base pair with its almost complement. Sequences on the end can have some effect, but a mismatch in the middle can be catastrophic. Correct. So in our summary here, the synthetic oligos behave according to the same rules. They need, salt help favors hybridization. Urea, formamide don't, but we have the additional and GC content affects it. But the shorter also will have the phenomenon of breathing at the ends that can destabilize the molecule, and that mismatches in the middle of the molecule can prevent stable hybrids from forming, or cause them to melt off as soon as they do form. So we're going to lose that cooperativity of bonding that we get with long molecules, so the short oligos don't behave quite the same way that longer molecules would do. Okay, this is a very old example, but it's a very good one and very telling example, and that's the use of allele-specific oligonucleotides to make a DNA test to look for the sickle cell allele of the hemoglobin beta locus. Okay, so in this mutation, there's a single nucleotide change, which leads to the change of one amino acid in the protein. Right, so that's the mutation we're going to be looking for, and we're going to use a single-stranded oligonucleotide with that mismatch, and also control without the mismatch, to basically detect alleles in that person's DNA with a single little nucleotide that's only 19 nucleotides long. So what's going to happen here, this shows your target DNA is going to be the genomic DNA from a person, and it's really much longer than what's shown here. It could go on for thousands of nucleotides. But of relevance is the probe, which is short, longer than what's shown here, but not a lot longer, which is actually, in this instance, fluorescently labeled at the end, so we can see it later. Now, in the very old examples, instead of fluorescence, it would be radioactive, it would be a P32 label. But the idea is the same. We're going to get our target DNA, we're going to mix it with our probe, and the probe is going to find its complement in the solution or on a filter, hybridize to it, and then we'll be able to see where it went by looking for the fluorescence or radioactivity, depending on the test. So the little yellow dot indicates that the molecule has been tagged, so we can observe it. Okay, so this is just one example of an allele-specific oligonucleotide, and it's been set up to detect this particular mutation. And the premise is that exact matches will hybridize and therefore be seen, and the inexact ones, which differ by a single nucleotide, will not form a stable hybrid, and that difference between those two will enable us to tell the genotype of the person. Okay, so the way these were done really is, first of all, you get the DNA sample from a person, and then for convenience, don't worry about how this is done right now, you actually PCR the region of interest to get a higher concentration of the DNA that's going to contain the mutation you're interested in. So we'll get to that next time. So the DNA from the PCR product is denatured, fixed to a filter paper, so it's stuck there in single-stranded form, so it's now available for hybridization to the oligos that we're going to use. So then we're going to design and label two oligos, one that exactly matches the wild-type sequence, and one that exactly matches the mutant sequence. And it can be either strand, as we'll get to next time. Okay, then we're going to hybridize them under conditions where only perfect hybrids will be stable, and then we can see which DNAs bond to which oligos, and therefore get the good genotype of the people. Okay, so here's just one version of the sequence of interest. Okay, so the wild-type allele has a GAG codon at position 6 in the protein. The mutant allele has a GTG. So those two alleles differ only in a single nucleotide, and that's what we're going to use to try to detect the correct match on our filter. We're going to use oligos are going to be 19 nucleotides long. So the point is, again, the mismatch will prevent stable hybridization, and that will allow us to distinguish the genotypes of the people. So here's a picture of one such filter. We have the DNA from 6 different people in spots, 1, 2, 3, 4, 5, 6, along the length of this filter paper. And we made duplicates, one for each of the oligonucleotides. Same DNA spots for the 6 people along the length of the filter. The A refers to the fact that this filter was hybridized to a radioactive oligonucleotide 19-mer probe that exactly matches the wild-type allele. So that oligo is expected to detect the wild-type allele in the person's DNA if it's present. The second filter, labeled S, is the one that matches the mutant allele, and so that's going to detect the presence of that allele in any DNA, any person that has it. And so this is shown schematically on the bottom, where you've got your two the targets on the bottom, and you've got your oligo, which is not the exact oligo I showed you before, but it contains it. You've got your T at one position, showing the exact match, and that will stay stable. And then, but that same T is going to conflict with the other allele, and that will not stay stable. So the pattern of spots tells us the genotypes of the people. So person number 1 hybridizes to the A allele and not to the S allele. Other people that have the A allele are people number 3, 4, and 5. People that have the S allele are 2, 3, 5, and 6. So simply by hybridizing to those two oligonucleotides, we can tell what the genotypes are of all 6 of the people. Right, so the A means it was wild type, S means it was beta S, and the filters are identical, right? What's different is which oligo was used for the hybridization. Okay. Person number 3 is homozygous for the wild type, heterozygous, homozygous for the beta S allele. Heterozygous. Their DNA hybridizes to both of those oligos. So they've got one of each, one from mom and one from dad. Okay. So we can go through all these and genotype them as far as who's got which. If you're homozygous, heterozygous, or homozygous for the other allele. Okay, so putting this in the context of hybrid formation, etc., we can see that the hybrid formation and stabilization was prevented by that single nucleotide mismatch. So by simply hybridizing these little oligos to the DNA of the different people, we could tell what their genotype was. Now, if you think what would have happened if this weren't done perfectly, if you had lower stringency, where the mismatch was less important, you might let the two might actually hybridize to each other, even though the sequences don't match perfectly, because that can happen in a very, very high salt condition where the mismatches are not quite as important. If we had done it too high stringency, it's only 19 mer, you've got breathing and all that other stuff going on. If it was too stringent, you might have melted off the perfect matches, because the length is only 19, which is not very long. So it's a very definite balancing act to get these things to work, but it tells you the power of understanding something about hybridization and what it can do for you. So here's my hemoglobin sequence handout. I'm going to put this off for a minute and just talk briefly about PCR, and then we're going to come back to this in a sec. Okay, so PCR reactions also use oligonucleotide primers, which are made by a machine. And of course, they're going to be synthetic, and they start off being single-stranded. And what they do is they are the start points for DNA synthesis reactions that are carried out by DNA polymerase. So even though they themselves are not made by polymerase, they're able to provide polymerase with a place to start replicating. Okay, they're specific to where we want the reaction to go, and we need two primers, one for each strand. Right? They also have to be specific in binding the DNA at only one location, and importantly for the current context, the correct annealing temperature to use in the PCR reaction is going to be determined by the melting temperature of the primer and target hybrid. You've got to get your melting temperature right, you've got to get your annealing temperature right in order to get good specificity in a PCR reaction, for the same reasons we talked about a minute ago. Okay, so remember PCR, we've heard of PCR, we're going to do it in more detail next time. You have a target DNA and you want to synthesize more of that target DNA. And the key ingredient here is the primers, the green primer for one strand and the red primer for the other strand. And this illustrates that the primer is going to bind at a specific location on one of the strands, and the other primer binds at a specific location on the other strand. And that DNA polymerase can then extend those primers using its normal DNA synthesis reaction to make new copies of that target DNA sequence. All right, so typically 55 degrees is used for annealing temperature, but that can vary quite a lot depending on what your situation is. So the outcome of a PCR reaction, the first few versions of it, clearly you need primers at every last step along the way, and the ability to do that eventually gives us tons and tons of the target DNA of interest, which will enable us to analyze or do other experiments. A student does a PCR reaction, does not get any product. Which of the following can explain this sad result? Annealing temperature too high, annealing temperature too low, one or both of the primers does not match exactly, A and C can both be true, B and C can both be true. All right, so the answer is D, A and C can both be true. Annealing temperature too high would mean that the primer, when it tried to bind to the template, would either not bind at all or melt off as soon as it did. So that would clearly get rid of any PCR product at all. If you can't bind your primer to your template, you're stuck. One or both of the primers does not match the template exactly. Well, that could be a problem also because the melting temperature might be different, and whatever annealing temperature would work for a perfect match might not work for something with errors in it, or mismatches in it anyway. So both A and C are true. A is extremely true, and C is less true, but could also actually happen. Okay, a student does a PCR reaction and gets several products of different sizes. Which of the following can explain this also sad result? Annealing temperature too high, annealing temperature too low, one or both primers does not match the template exactly, A and C can both be true, B and C can both be true. Well, I vote for E. Clearly, annealing temperature too low means that the primer could bind nonspecifically at other locations in the genome, and therefore cause the formation of a product other than the one you intended. Also, one or both of the primers does not match exactly. It might match somewhere else and also cause problems for that reason. Okay, we're only going to do part of this now because the rest of the discussion is going to be a little more complicated. Our first question is going to be to design a oligo that's going to allow us to detect the wild type and mutant alleles of this particular part of really the hemoglobin locus. Okay, so what you're seeing here, and this is kind of confusing, it's a DNA sequence. You can see there are T's in it. It's single-stranded. Only one of the strands is shown. Okay, and what this is a portion of what's known as a cDNA or copy DNA, where it was the DNA that was actually made by starting with a mature mRNA and converting it enzymatically to DNA by means of reverse transcriptase. So most of the sequences that you see in GenBank or anywhere else are going to be in the DNA form because they've been cloned and they've been sequenced and they're basically stored in that form because RNA is not stable and cloned DNA is stable indefinitely. So even though it's an RNA sequence officially, it's really in the form of a DNA where you've got T's instead of U's. That can be confusing, but that's how it is. The fact that it's single-stranded is also confusing because they're only going to show you the one strand, which is a strand from which you can actually read the protein sequence. So the other strand's actually present normally. It's just that they're not showing it to you because because of complementarity, you can, if you know one strand, you certainly know the other. And it's more convenient to show the strand where you can actually read the protein sequence. All right, so this is one strand, part of one strand of DNA that's actually a replica of an mRNA, and it encodes the first part of the beta chain of hemoglobin. And the ATG is underlined, and the T, which is of interest, is the one that we're going to be looking for. Okay, that would be the sickle cell allele. The wild type would read GAG, and this allele reads GTG. So the first thing we're going to try to do is just design an oligonucleotide, a 20-mer oligonucleotide, that's going to allow us to detect either the T or the A at that particular position. Okay, so part A is to write the sequence of a 20-mer that will detect the wild type. Part B is to write the sequence of a 20-mer oligo that will detect the mutant. All right, so B is easier. Let's do that one first because that's the sequence that we're given. We don't have to mess with it. Okay, now don't make this hard. All right. So the T that we're looking for, we're looking for the T, but we need a 20-mer oligo to hybridize to the sequence in order to be able to detect it. So what you do simply is complement the sequence that we underline. A 20-mer would say 10 on either side of the T approximately, and so that's the total length of 20. And then to detect the sequence, you need something that's complementary to it so that will hybridize to it. So if you then label it, you'll be able to see it in some manner or another. Okay, so that's the underlined section is what we need to make our 20-mer complementary to. So I just wrote out the T goes with the A, C, T, G, etc., reading the complement of that strand. And then, of course, remember that if you're going to be complementing something, the two strands of the DNA are anti-parallel, and the top strand is written 5' to 3'. The oligo that you write is got to be opposite. It's going to read 3' to 5' as the complement is read from the sequence. So that's the answer to part. I'm giving answers to worksheets. Oh well. Okay. And the wild type allele is the same idea. All you do is you take that T and change it to an A to make it the wild type allele. And then you just do the same thing. You just complement each nucleotide as they go along. And then, of course, reverse the polarity because of the anti-parallel nature of the double-strand DNA. Now, the next part having to do with what would detect the RNA, we're going to deal with that next time because then you have to consider the fact that actually both strands are going to be there in any DNA from a person. And so sometimes you have to pick which strand you want to look at. But we'll deal with that next time. End.